Hello everyone, I am Sibgha Batool, PhD scholar of Department of Pharmacy, Quaid-i-Azam University, Islamabad. I am very much thankful to the organizing committee for giving me an opportunity to present my work in this international conference. Today, the topic which I am going to present is macrophage targeting for the treatment of cutaneous leishmaniasis using novel technology and drug delivery approach. First of all, the introduction. Leishmaniasis is a vector-borne protozoan infection which is caused by the Leishmania parasites, and it is transmitted by the bite of the infected female sandfly. More than 20 such species are involved in causing this infection. In the life cycle of the Leishmania parasites, there are two stages. One is the pro-mastigod stage, which is the motile one, and other is the amastigod stage, which is non-motile one. Among all the forms of the leishmaniasis, cutaneous leishmaniasis is the most common one, in which ulcerative skin lesions are formed at the site of the sandfly bite. So how it occurs? The sandfly transfer the infected promastigods, which permeate into different layers of the skin, and they are engulfed by the macrophages, where they are converted into the amastigod stage, and then they divide and redivide to infect different cells and then increase the risk of the infection. Now, the rationale of my study, there are many different medications that are already available for the treatment of cutaneous leishmaniasis, but there are some problems that are associated with those medications. So today I will talk about miltefosine, which is HEPC, and it belongs to BCS class 3, so it has low permeability, and there are some problems that when it is given orally, it causes the gastrointestinal toxicity. Similarly, it causes hemolysis with the IV therapy. So according to WHO, topical drug delivery is the recommended route for the treatment of cutaneous leishmaniasis as compared to other routes. So we selected transfersomes because they are biocompatible, they are highly deformable in nature, they are site specific, and they control the release of the drug. Next is the mechanism of penetration. The driving force required for the transport of transfersomes is basically the difference in the water content, and this is due to the osmotic gradient between the dry skin and hydrated epidermis. So as you can see in the figure, they can pass through narrow constriction that is 5 to 10 times less than their own diameter. The formulation which we developed had the particle size less than 200 nanometer, and zeta potential was negative due to the negative charge present on the lipid. And you can see in figure A of the TEM analysis that the particles are spherical in shape and they are homogeneously distributed. After that, in vitro release study was performed at pH 5.5, and you can see in graph B that almost 90% of the drug is released within 4 hours, and in case of HePCTG, which is our formulation, the drug is released in sustained manner, and the overall pattern of the release follow the frequent diffusion. Next is the ex vivo skin permeation study, and this study was performed because, as I told you earlier, that miltefosine has the permeability issue. So you can see in the figure 2A that transfersomes, dye-loaded transfersomes penetrated into deeper layers of the skin very easily, and so we can say that the permeation could be enhanced by incorporating the drug into transfersomes. However, we needed the sustained release of the formulation, so the transfersomes were further loaded into the gel. After that, we performed the in vitro anti-leishmanial analysis, and in graph B, you can see that the percentage inhibition for HePCT is greater as compared to HePC solution. So we can say that the percentage inhibition of HePCT is higher against the leishmanial parasite as compared to the solution. Similarly, we performed the cytotoxicity assay, and we found that the cytotoxicity of the solution is greater as compared to our formulation. After that, macrophage uptake study was also performed because it was the main objective of my study. So it is clearly evident from figure D1 that the macrophages uptake the transfersomes and they produce the fluorescence as compared to the control one. Then we performed the skin irritation study and histopathological study, and it was observed that the positive control, which was 0.8% formalin, produced the toxic effects to the skin, and the epidermis became thin and loose texture was observed. While in case of gel, there was no toxic effect, and it proves that the gel was quite safe. After that, in vivo anti-leishmanial assay was performed, and it was observed that the lesion size was reduced in case of HePCTG, which was our formulation, as compared to the untreated and HePC gel. So here I conclude that transfersomes are the potential drug delivery systems for topical delivery of anti-leishmanial drugs at their targeted sites, resulting in better permeation, controlled drug release, enhanced cytotoxicity, and improved anti-leishmanial efficacy, most particularly for the treatment of cutaneous leishmaniasis. Thank you so much for your attention.